My name is Bob Schwartz, and I serve as the Director of Special Projects for MU Extension's Business Development Program. I also work closely with the Missouri Small Business Development Center, and it's my honor to host today's webinar. Before I introduce our speaker, let me review a few brief housekeeping details. All participants will be muted during the presentation. If you have questions for Dr. Singh, please submit them via the chat box, which will be monitored. I'll be happy to share those with Dr. Singh at the conclusion of his presentation. With that, it's my great pleasure to introduce our speaker today, Dr. Kamal Singh, Associate Research Professor at the University of Missouri-Columbia, where he also serves as the Assistant Director of the Molecular Interactions Core. Dr. Singh is also an associated faculty member of the Department of Laboratory Medicine at the Karolinska Institute, which is located in Stockholm, Sweden. Dr. Singh received his BS degrees, yes, that's plural, in physics, chemistry, and mathematics from Agra University in Agra, India. His MS in physics, also from Agra University, and his PhD in quantum physics from Banaras Hindu University in Varanasi, India. Dr. Singh is also a trained medicinal chemist. For about 25 years, Dr. Singh's research has been focused on understanding the molecular mechanism of nucleic acid metabolism by bacterial, viral, and mammalian replication enzymes and drug discovery against a variety of pathogens. More specifically, the primary focus of his research is the discovery of drugs treating viruses and cancer using computer-aided drug design, as well as biophysical and biochemical techniques. For the past 11 years, Dr. Singh's research has been mainly focused on severe acute respiratory syndrome coronavirus, also known as SARS-CoV, Middle East respiratory syndrome coronavirus, also known as MERS-CoV, human immunodeficiency virus type 1, or HIV-1, foot and mouth disease virus, hepatitis C, and also hepatitis B viruses. Dr. Singh is the author of numerous publications, and his lab has been granted 4 US patents, 2 of which have already been licensed. I would also note that he serves as guest editor for the International Journal of Biological Macromolecules, and he served as guest editor for the journal Cellular and Molecular Biology. Dr. Singh, thank you for being with us today. With that, we look forward to your presentation.
Well, thank you so much, Dr. Schwartz. It's a pleasure to be here. And before I start my seminar or presentation, I have to share the screen, which I will struggle, of course, as always. Share a screen. Let's see, desktop 1. And on this desktop, I will share the PowerPoint. Well, one thing I want to tell about my education, about Agra, and I would like to point out here, Agra has a monument, and the only monument in the world built for a woman. There is no other monument in the world that's built for a woman. So that's called Taj Mahal, excuse me. I was there 4 years, and I only went once. That's how much time I had. In any case, I have been in MU, as Dr. Schwartz says, for last 11 plus years and have been working on different kind of viruses. Even though from my education, you can see that I'm not a virologist. I graduated as a physics person, but somehow things changed, and I'm here presenting the virus-related work. So thanks for the opportunity. Let's get into the business. So what I want to do is present some background before we go in detail, because I think some folks may not know exactly what coronaviruses are and how they have evolved. I won't go too much details in science, but overview. So coronaviruses have been known to infect respiratory and gastrointestinal tracts. A lot of times self-limiting, means like common cold. Body clears it after a while. Um, Come on. I'm sorry, I'm struggling with my mouse. Okay, these are not new actually. Coronaviruses have been in existence for quite long, and very first one was discovered somewhere in 1965 and 66. But as I said, they only cause common cold, so there weren't not much attention was paid to them. In fact, actually, I read somewhere that there was a flu-like epidemic in 1890s in Russia, and that was related to coronavirus. So even though they were discovered a lot later, but first deadly known coronavirus emerged in 2002, 2003, and then MERS, Middle East Respiratory Syndrome coronavirus emerged in 2012. And these two coronaviruses, they emerged, and after a while they were contained. MERS still is there, but not as deadly as the new one. So the recent coronavirus, SARS coronavirus 2, emerged in Wuhan, China, early December, and which is the cause of COVID-19. COVID-19 stands for coronavirus disease 19, and 19 comes from 2019. It definitely is not 19th coronavirus. It's basically, if you think of human coronavirus, it is 4267th coronavirus. So not only in humans, coronavirus also infect animals. So that way, one can say coronaviruses are zoonotic viruses, means they transfer between human to animals. And common ancestor, as you see here, is not known yet, but bats and some birds are hosts for this coronaviruses, and they can be transferred to, let's say, mice to humans, and different kind of animals, camels, dogs, pigs, cows, so on and so forth, including fish. And it happens because one animal depends upon the other one, and if one animal eats the meat or gets in touch with another animal, the virus gets transformed. I don't want you to remember all these alpha, beta, gamma, and delta coronaviruses. These are only for scientists. The only thing I want you to know that the newest coronavirus, which is SARS coronavirus 2, belongs to lineage B, which has not much meaning. But here, one thing I want to point out. We do say it's a coronavirus, SARS coronavirus 2, but when you go to see and try to analyze what is the closest virus to SARS coronavirus 2 or COVID-19 causing virus, and what you see here is one of my colleagues did this analysis early in March, and actually sometime in mid-March. So what he did, he took the sequences, sequences started being available that time. He took the sequences and then aligned them. What you see here in black, each single dot is a different virus. Clearly, black lines, blue lines, green lines, and red are dark orange lines. They represent different viruses. Of course, this virus came from China, but that's not what we want to talk about here. What we want to talk about is, or discuss about is, what animal virus is closest. So when we align the sequence, you see the bat coronavirus right here on the top. That's the closest. So somehow, some way, some unknown way, this coronavirus, the current coronavirus, has come from a bat coronavirus. And we still call it SARS coronavirus 2, which is not closely related to it. In fact, if you see from blue to green is closer than orange. But for convenience, we do call it SARS coronavirus 2. And that's how we all know it now. In fact, it's close to a bat coronavirus that, uh, and that bat coronavirus actually is also found in China. Now let's see the structure, what coronavirus looks like. You probably have seen this kind of picture in the right a lot. And but you see this red spike sticking out. And the name coronavirus actually comes from these red things sticking out. So corona, which stands for crown, and if you see the virus as such, it looks like a crown. So you have these different proteins. This is enveloped virus, means whole the genome is packed into a shell, and that shell contains these proteins. So SARS spike protein, glycoprotein, envelope, and then other different protein. What I want you to focus is the spike protein that we will talk about, and almost every single vaccine today is focused on one way or the other on this spike protein. And then what you see here is this blue spiral kind of thing. This is actually genome. And if you take it out and stretch it, it stretches about 30,000 bases, means 30,000 units of RNA. So, we know that AUGC are the units. And this genome of coronavirus, when it translates, it doesn't come with any protein, by the way, itself. Every single protein of coronavirus is made by the host cell after the infection. So it codes, the genome encodes around 25 to 26 proteins. Still, there is some confusion about it. Of those 25 to 26 proteins, 16 participate in replication, means making the copy of the genome, and rest of them participate in packing the virus. And of those 25, 26 proteins, the focus of drug development has been on two. One is the spike protein, obviously for vaccine purposes, and one is this right here, RDRP. RDRP stands for RNA-dependent RNA polymerase. And one of the drugs you might have heard really a lot called remdesivir. And remdesivir actually does target this RDRP. We'll talk about it a little bit more. So as we will talk about the spike protein. So as of yesterday, and I forgot to give the site. This slide has been taken. Let me see where the site is. I will send it to you at some point. As of yesterday, there are 319 treatments. None of them are available for general public, but they are investigational drugs, and 213 vaccines are in process of development. And of those 213 vaccines, which will be focus of today, they have been developed using 9 platforms, 9 kind of categories, and 36 vaccines are in one or the other phase of clinical trials. And those phases are phase 1, when you have limited, let's say, patients, you test them. Phase 2, you have more patients. Phase 3, you have thousands of patients. And then phase 4, if you want to call, is a regulatory review when FDA reviews for the safety. So 36 of them are in that process as of yesterday. So let's go for vaccine strategies. So what you see here are the 9 different kind of strategies. So I will start from the bottom left. Attenuated virus, I will come to that point, then virus inactivated, and then they're called viral vector, replicating viral vector, and non-replicating viral vector. Then a spike protein, either whole or part of it. Then RNA in lipid nanoparticles. Nanoparticles are small sphere-shaped particles, and one can put the genome within there. And then DNA. So the difference between RNA and DNA vaccine, they more or less have same information, but RNA is a fragile molecule, so it cannot be given as such, as opposed to DNA, which can survive in the nature for years, years, and years. So it can be given as such. In some long DNA, you have something that codes one of the viral protein. Now, we all know vaccines are for prevention. They are not treatments. So whenever you give vaccine, you have to wait for a few days for immune response to develop. And that immune response actually protects us from the disease. The other, here you see VLP is virus-like particle. We have the part of the protein or whole protein of coronavirus in this case, and you embed into some kind of organic or inorganic molecule, and then you inject that. And surprisingly, there was another vaccine, hundreds of years old, BCG vaccine, actually that is given for tuberculosis, and it has shown some effect. So one Australian company is working on this. So we'll go over all these platforms and how many drugs are in the advanced stage. We are not going to talk about everything because we'll take forever to go through them. So traditional vaccine development takes a lot of time. First, you identify the target, then you do exploratory studies and lab testing, toxicology, then you submit the new drug invention protocol, then carry out phase 1, phase 2, phase 3, and regulation, so on and so forth. It takes around 15 years to develop. So that we can call a 4x4 100-meter relay compared to COVID-19 specific, which was, you can call 4 into 100-meter relay, because many of the processes have been merged. For example, here, right in the middle, phase 1, phase 2, phase 3 as kind of combined, and companies were giving permission to include all of them in one or two studies. So the knowledge already existed from SARS coronavirus 1 and MERS coronavirus. We used that knowledge, developed the toxicological studies. Actually, that also kind of new was known. IND was submitted, and the race started for developing the vaccine. So let's say early January, not early January, I'm sorry, sometime late January to February, these processes started, and we were supposed to get results by, let's say, November. None of them have come yet out, but hopefully early next year, we will have these vaccines out in the market for public use. There are a lot of bottlenecks they have to go through other than development. So inactivated virus. So all these platforms, I'll go briefly about them, what they are, and how these vaccines are created. Inactivated virus is a simple virus. You take virus and you inactivate with low energetic, either electron radius or neutron radiation or X-ray radiation. What it does, it actually destroys its genome. So the virus is now inactivated in the sense that it cannot replicate itself, only it can enter to the cell. It can enter to the cell, it can't replicate, cell will recognize, our host will recognize these spike proteins and develop the immune system. So there are 14 candidates right now in advanced stage. And those are 3 in regulatory review in China, 3 are in phase 3 in India and China, and 1 in Kazakhstan. Now, just as an example of this kind of platform is polio and flu. The flu vaccine that you take, or we take, is basically this inactivated virus, influenza virus. Then we come to attenuated virus, means virus can progress. Inactivated virus cannot progress. It goes there, immune response comes up, and that's it, done. Attenuated virus can replicate, but it replicates very slow. So what the strategy is used is You engineer a virus, means some of the genes are inactivated, and then you take a virus that is a regular one, you petri dish them, in a dish, grow them, take them out. So some population of those viruses have mix of the two. And that population is actually called attenuated virus, which is eventually used for immunization. There are 4 candidates right now in the making. All of them are in phase one trial, clinical trial, and the examples are measles and mumps. Actually, vaccinia virus, to be exact, is one of the examples. Now, the 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 bottleneck in both inactivated virus and attenuated virus is production of virus. How much virus you can produce, and can you meet the public demand? As of now, we are struggling. Everybody is struggling. For example, this coronavirus 2 is uh, is is uh, is produced in Vero E6 cells. Those are not even lung cells, which where this virus, in fact, they are kidney cells, and the yield is not that high. So that is going to be a problem. So it's a logistic problem, and some way or other, companies have to solve it. So to solve that problem, they say, okay, well, if that's the bottleneck, let's move on to next different strategy, a different platform. And all these platforms here onwards are related to this protein that you see on the surface of the virus. It's called a spike protein. And there is one portion of this spike protein is called a receptor binding domain. That's the portion actually very first time when virus attacks us, that green region is the one that attaches to the cell. And where it attaches is actually a protein, a surface protein called ACE2, angiotensin converting enzyme 2. And just for the reference, ACE2 inhibitors are given for heart problems. So that's where it attacks. So what companies are trying, they are trying one or the other portion of this protein, expressing them, making the protein or peptide out of them, and using them as a vaccine. So once they inject it in the body, what will happen is immune response will be activated. And once when the virus infects the cell, there is already immune response. So it will not be, the cell will not be infected. And this category actually right now has 72 candidates in making. There are 4 in RR, means regulatory review, that in India, China, and Japan. 4 are in phase 3, and that also includes actually USA. And 2 countries actually already have approved. Russia and China have approved um, 2 vaccines, their own vaccines. I don't remember Chinese vaccine name, but I do remember Russian vaccine name. I think they called it Sputnik, Sputnik 1 or something like that. The examples of this kind of vaccines are, from the peptide or from the protein of the virus, are hepatitis B. A lot of us take them all the time, and shingles. Then you have DNA-based vaccine. As I mentioned before, you have DNA, just put a gene in a long DNA and just use that as a part of vaccine, inject into the body, and what it will do is the body will take this DNA and make the protein out of it, and that protein will, because it's outside protein, the immune response will be triggered, and that way you will have immune response when virus attacks. There are few in phase 2 and 3 trial. They haven't gone to complete 3 trials. And again, USA, China, Japan, and India are leading in this. The sad part of this is there are 17 candidates. That's not sad part. The sad part is until today, no DNA vaccine has been approved for the treatment of any virus. So that poses a kind of risk, and that is another bottleneck. We don't know what's going to happen to this foreign DNA when it comes to our body. Same thing is for RNA-based vaccines. There are 27 candidates, and the famous ones are Moderna and Pfizer. As you might have heard in couple of days back, there was news that Pfizer has released its phase 1 results. They are ready to release their phase 3 results sometime in late this month, but they are not filing, and that's kind of sad part. The filing for the approval to FDA until late November or so. The same sad part here is that nobody knows the safety of these RNA and DNA-based vaccines because there haven't been examples of them, and that's a little bit problematic. So I think these companies, whenever they release these vaccines, will have gone through entire safety process, and that probably is taking the time. We come to replication virus. So replicating viruses, sorry. So the replicating viruses have on the surface, these are different viruses for adenoviruses, for example. What these viruses have is they are engineered such a way that they have a spike protein on the surface and part of genome in the virus, replicating virus genome, you can say carrier virus. And that what happens, it continuously replicates in the cell. It doesn't cause the disease because you need entire virus, but it does create some proteins within the cell, within the host, so that the immune response is triggered and it's ready when the real virus attacks. So that's another one. And the next one is non-replicating, and I'm sorry, I missed it. There are 18 candidates right now. Ebola and dengue vaccines are based on this technique. None of them is in advanced phase. Then comes the non-replicating virus, means you still have the similar kind of situation as you had in the replicating virus. And when I say replicating, it's kind of scientific term, but replicating means producing the progeny, means the daughter virus. That's what it means, replicating virus. So non-replicating virus, because the gene is kind of broken, and you have the spike proteins, of course, on the virus, the carrier virus. So that does not replicate yet. It serves as a carrier. It takes these proteins easily, but does not replicate and triggers the immune response. And this is actually, there are 26 candidates. Of these 26 candidates, 6 are in regulatory review. This is wrong here. USA shouldn't have been there. Nothing is in regulatory review in USA, but there are 6 candidates. They are in advanced stage, and some of them are in USA, UK, India, China, Canada, and Germany. And some examples of these are Janssen, that has been paused, and famously, you might have heard, AstraZeneca has paused its clinical trial, and that was based on non-replicating virus. There has no vaccine yet approved that uses this technology to develop, or this platform to develop a vaccine. The final 2 are virus-like particles, for example, nanoparticles. Nanoparticles have surface proteins on it. They are easily delivered. There are 16 candidates in the making right now. The example is human papillomavirus vaccine is actually based on that. And this is a weird example, or unusual example, I should say. If studies have shown, an Australian study actually, shown that BCG vaccine is quite effective against coronavirus. So there is only one repurposing vaccine is in the works, and that is done by Murdoch Children's Research Institute in Australia. Examples haven't been, no vaccine has been yet repurposed for any viral infection. So that's the summary here. If you take this whole thing as a syringe right here in the left, and different phases and different kind of vaccines, a lot of progress has been made in last, what, 10 months at the most, not even 10 months, 8 months and 9 months. And hopefully we will soon have the vaccines. So you saw the example, most of these vaccines are almost everything except inactivated and live attenuated virus is focused on spike protein. So lately there have been a lot of talk about the spike mutation, and that's where I will start my work and the risk what these targeting spike protein may pose. So there is a mutation has been talked a lot called, actually this typo should be D as Dave 614G, like typed here. So if you search for in Google, within less than a second, you hit around 2 million um, search articles. That tells that, well, the SARS coronavirus 2 has been mutating, and one of the prominent mutation is this D614G. D stands for aspartic acid, G stands for glycine. So one amino acid residue, it changed from aspartic acid to glycine. So that's when I started talking to one of my colleagues in radiology. Actually, to be exact, it was May 6th. And I was on Como, and right after my interview, this gentleman called me and started talking about my work and on coronavirus and congratulating me. So I inadvertently I said to him that, well, if I had a student who could do a lot of programming, that would have been good. And he said his son, who's in 8th grade, does a lot of programming, and he will introduce to him. And same time, Austin Spratt, he was working in the lab, but because of the ramp down of the research, he couldn't work in the lab. So I asked him, Austin, do you know any programming? And he said, yes, he does. So I put these two brilliant students on the work. And these students gave fantastic results, which I will go over, but a little bit detail about, again, I'm sorry, I apologize. This should have been D, not G. It's a copy and paste error. So this is how the genome of coronavirus that encodes spike protein looks. So there are different sections. This is receptor binding domain. That's common right here, as I showed before. But what happens is this protein is cleaved as soon as it gets to inside the cell, right here in 2 parts, and the job done. That's all it does. It attacks the cell, gets inside, and done. And this mutation, when the structure was solved, could be seen that this aspartic acid right here has interactions. And these interactions are meant to stabilize the whole thing among different parts of the protein. I will not go what kind of interactions, but once this mutation happens, D to G, these interactions are lost. So this mutation, and when someone did work in Los Alamos National Laboratory, she, to be exact, her name is Betty Corber. So she reported that the virus containing this mutation is more infective than the wild type virus. Wild type virus means the one that came from Wuhan, China, or the reference virus that you can say. So I put these students and I said, okay, why don't you start working on this? And they came up with the results, which I will show in a second. But this study did make headlines, and it was on our news release in MU. So what was the study? I told Austin, why don't you look the genome of the virus in the USA? Because the problem was that the virus is replicating in the USA relentlessly. Why? So I said, why don't you go look? Where are the mutations compared to the virus that comes from China? Wuhan, basically, that's called reference virus. So he looked at this spike protein right here. And you see that each dot here represents a mutation. So green sequences are from USA, The blue are from Europe. The red, kind of red, you can say light red, are from China. And the orange inner circle is from Indian sequences. So he came up with, with this result and said, well, what we see is not only this mutation, there is other mutation that consistently exists, at least in all USA sequences. I said, okay, well, then keep going, do some more research. And so asked Satwik, who was 8th grader, he took the challenge and started looking for what other mutations do exist in the virus, especially in the USA. And he came up with the idea. And actually, naming them doesn't mean much, but one mutation is in that gene, P323L, means proline to leucine, is in the gene that targets that is targeted by remdesivir. So what Satwik came with the results that all these mutations coexist, and by end of June, around 92% virus in USA has 3 mutations consistently. That was fascinating, and so they still kept working. And by August 3, every single virus in USA has these 3 mutations. So we proposed that time, okay, well, so it's not, the story does not limit to this only mutation, B614G. It is also, there are 2 mutations, they coexist with them, virus. And that is probably the reason virus is more infectious compared to viruses, let's say in China, where we could find only 3 sequences that had these mutations. Well, there were some other mutations came up and went. Some of them were mutually exclusive. For example, there was a mutation here. It was highest around somewhere in 52% in February. But as these mutations evolved, shown here in the left, these mutations started disappearing to almost 0. So we submitted this article and was highly appreciated and recognized, and got published paper in actually 3 days. So what you see here is the mutual correlation, means how many other mutations are existing there. We only took certain genes, we did not take entire virus. Right now, the work is ready to be published actually for the entire virus. Then we define the mechanism, what mechanism could cause the higher infectivity of the virus that has these mutations. And again, this is part of science. The work is going on right now in the lab. We have these proteins, we have made these mutations, and it looks like from preliminary results that the prediction is correct. So Austin kept working on it, as I said before, and he looked for entire viral genome. So at RNA level and at protein level. So why I'm emphasizing here RNA level? Some of the vaccines that are developed are RNA molecules. So if there is, let's say, a dot mutation that does not match with the RNA molecule that is given as a vaccine, apparently it's not going to work against this virus, or this virus right here. So these things that do pose limitation of vaccines if and when these vaccines are available. So here in the left RNA level, in the protein level, and what it tells in nutshell, you see a lot of dots. What it tells is that each dot is a mutation, and virus has a lot of mutations. So the circulating virus in the United States and in the world, actually for now, except China, because we don't have many sequences from there. So that's our limitation, we can't analyze them. All these mutations in this green area, that the green strip you see, they will cause at some point one or the other problem, either for drug development or for vaccine development. So we have to think these strategies that can circumvent these mutations. These are some other mutations of same kind. There is another protein that has a lot of mutations, especially in USA population, which is here in the green. I don't know why he changed the sequence of it. So the idea is now we know the virus is evolving, the virus has mutations. So what can be done? And people are, we as a scientist, our goal is to put out the information and let companies think about how they are going to circumvent, or we ourselves, if we get, let's say, funding support from some federal agency, we might ourselves take up that challenge. In the meantime, what are treatment targets? We already talked about vaccine targets. So early in the time when coronavirus was at very high rate in the United States, you might have heard a lot of talk about chloroquine and hydroxychloroquine. So what this figure shows that, okay, the drugs are existing in the world. Can some of these drugs be used? This is actually what this figure shows is the life cycle of the virus, which I'm not going to go through, but these different, uh, you can say, stop points or pauses tell virus came here, attached here, and then got inside the cell. Then genome was opened, and this genome was translated into proteins. And at different steps, wherever you see the red signal and the drug written there, those drugs can be used. For example, this protein right here at the surface of the cell called TRMPS2, it cleaves that spike protein S1 and S2 for virus to be infected. Now, if we can block TRMPS2 by these two drugs, and that will be repurposing, then the infection virus will go inside, but will never be infectious. And then we can block here the endocytosis or acidification of the virus that releases the genome by chloroquine and hydroxychloroquine. And apparently this technique didn't work. The clinical trials did not show any effect of either chloroquine or hydroxychloroquine on the virus replication. So there are other mechanisms are involved, which we don't know. But then HIV drugs, leponavir and ritonavir, and another drug, disulfiram, that is also for different virus. I don't remember which virus it was. That can they be used as protease inhibitor, which cuts the viral proteins into mature proteins, or remdesivir, favipiravir, or ribavirin? Can they be used to block the replication of the virus, or cyclosporin A can be used to block some kind of pathway that virus hijacks? So some of these drugs we ended up testing. This was the hypothesis. So what we tested was, we'll show in a couple of next slides. First of all, remdesivir. We proposed remdesivir in late April that there are 4 drugs out there. And this article was actually highly accessed. It made to the cover of the journal. So what you see here this is the title of the article. That doesn't mean these are all collaborators, including myself. Remdesivir looks like this right here. It's a prodrug. It is adenosine derivative because this portion looks like adenosine, one of the DNA bases. When it goes to cell, this drug is converted into this kind of form, which is called triphosphate form or metabolic form. That is accepted by this enzyme right here. You see in green, blue, and red, and here in those balls, ball mode, you see this is the drug. And we propose that, well, this drug can be used as an alternative while we get some treatment. So at that time, it probably triggered because we reported and there were a lot of talk about it. So this drug was given actually temporary authorization to use in few countries, especially in US, in Italy, and in Spain. But we also report 3 more drugs, and that was favipiravir, as you saw here, right here in this, favipiravir and ribavirin, as well as fluorouracil. So favipiravir is actually already a drug that is used for treatment of influenza in Japan. This drug also got temporary approval from Italy and Spain, Japan, few more countries. I don't exactly remember, India as well. Not in US. Favipiravir does not have approval to be used in United States. And actually the results of favipiravir clinical trials are really good. But what was stunning that last week or week before, the results of clinical trial of remdesivir came out. And in many, many interviews, I have mentioned that, well, I will be the happiest person if remdesivir works, but there is not much hope because from our work, we could say the drug does not bind to this enzyme as effectively as it should, or as effectively as other drugs bind. And this turns out to be true. However, it does show some effect, and that's what we have been. That's why this has been used and is still used for the patients who are on the ventilator and who are 65 or older. So it reduces the viral infection period. So there is still some effect. It's not optimal, but the efforts continue. So how we can repurpose other drugs? That was the strategy. We still are looking for drugs. No drugs are available. Virus is kind of out of control. I mean, it can be contained if we take safety measures. So to repurpose the drugs, we had to find out what kind of effects after virus gets into the cell does to the cell. There are many different pathways they are, they are activated or down-regulated when some kind of pathogen attacks the cell. So we had to find out pathways. So what we did, we took live virus from a Swedish patient. We infected the cells, virus-6 cells, kidney cells, and this work was done actually in Sweden, in Karolinska Institute. And obviously MU was collaborator on that. So we took the virus, we infected the cells, we took out the RNA, whole RNA from the virus, we isolated RNA, we isolated proteins from the cell, and we did a lot of proteomics and multiomics, you can say, the sequencing and all, and came up with the idea that you can see here, 24 and 20, 24, 20, 48 and 72 hours. At 72 hours, we could find 4 signaling pathways that were highly dysregulated. And these pathways are involved in different kind of signaling. Something attacks to cell and they give the message, and the cytokines or interferons, they get activated and they save from the disease. Not to go into too much details. So what we did, we chose few drugs. All those drugs are shown here. Vertaminin, MK2206. I mean, they're kind of cancer drugs. Some are other drugs here. And we say, let's say, if we can use any of these drugs that can be repurposed, they are already known, they are approved by FDA, if they can be repurposed for coronavirus 2 infection or COVID-19 disease. Of all these drugs we tested, one drug is called MK2206 actually did show significant inhibition of the virus replication in the cell. So, and this drug has been picked up by a clinical trial, and we are awaiting the results. So this has been our contribution to the drugs, but two things to the field of coronavirus, but two things I would like to point out here. One, we have to be careful about the mutation in the spike protein or in the virus itself. The virus that is circulating in the United States is highly mutated. We have to give consideration to that. We do have to continue the second part. We do have to keep continuing our efforts to develop more drugs. Towards that end, we have our lab in collaboration with the University of Nebraska Medical Center. We have identified 2 entry inhibitors, and unfortunately, I can't present them here because people are waiting for invention disclosure and many other different paperwork form. But having said that, what is an ideal drug? So an ideal drug can start, let's say, from the outer circuit circle, and slowly goes to inner circle, ideal vaccine. So you choose the platform, design target, 4, 5 targets, then prepare them in small batches, do the testing in the lab, select the target, then target validation, validate the target if that's the target. So again, that's done in the lab. Then take into the small animals. For example, the mice that express ACE2, because if an animal does not express the protein that binds coronavirus, it's useless. So we have to make the mice that express ACE2 protein, or use Syrian hamster, that's non-human primate, check in them, and then take into the phase 1 study, that's the inner circle. Then phase 2, where safety and efficacy, phase 3, efficacy and protection, and then keep going through that regulatory regulation. Then scale up the product. From there, you scale up the product, manufacturing, which is a bottleneck, then establish supply chain and the worldwide distribution. So this sphere kind of summarizes everything that one can do for vaccines, and it's not easy. There are many places where bottlenecks can come. In fact, both RNA and DNA vaccines, they have to be stored as -80 until they are being given. I mean, how can you or anyone can make sure that all these vaccines, these two vaccines, stay at -80 all the time, especially in low and middle income countries where power is a big problem. So these are things one has to consider. And from here, I will summarize. So many vaccines, as we saw, are at advanced stage. None of them are available for community use right now, hopefully early 2021, and that's being optimistic. No treatments. Remdesivir turned out to be a poor drug, not optimal drug. There are many mutations in the virus. They will cause problem, can affect vaccine outcomes. So our research must continue. From here, I will stop, but I can't stop thanking without thanking people who have been involved in all this work. My own lab at MU, Grace Young does a lot of protein purification, Anna does a lot of experiments. Austin and Satwik, I showed their work. Rohit was a previous student, so was Gilberto Perez. And I mentioned these two girls, they both are doctors now. I have collaboration, active collaboration in Rutgers. My mentor, actually postdoc mentor, Dr. Modak, collaborator, Dr. Suzuki. We are working on a different project. I have a patent license with Dr. Burge. That's on cancer drug. Karolinska Institute, Dr. Sonnerberg, just to emphasize his importance, not only he supports financially our lab, he is also part of Nobel Committee, Nobel Prize nomination committee. Any drug in European Union of HIV or any virus has to go through his desk. Dr. Ujjwal Niyogi is my friend and collaborator. We have many papers together. I have active collaboration with Dr. Sid Bera-Reddy. He is a well-known HIV person, actually has few papers with Dr. Anthony Fauci. Dr. Amar Natarajan is a medicinal chemist. Whenever I need some kind of modification in the molecules, I look at him. Dr. Carol MacArthur, UMKC, a lot of HIV work is done in collaboration with Dr. MacArthur. Here at my University of Missouri, collaborator, Dr. Chris Larson, Dr. Tom Quinn, Gary Weissman. We have gone together, and Dr. Krishna Sharma, we have gone together. Dr. McPatrish, we have gone together with him, and Dr. Zhao Heng. This is new collaboration with Dr. Adam Wali Connell in VA and Dr. Arur Anaya. And all these agencies, Karolinska Institute, I have appointment there, they fund my lab. And I'm sorry, I can't read what this is, but what it stands for is NIH equivalent of Sweden. So they support my research. Bond Life Science Center, I'm proud to be here, and I have been in the same office since I came here. They also support my research, National Institute of Health and American Lung Association. thank you for your attention. I will take any question from here.
Great. Thank you so much, Dr. Singh. If you have questions for Dr. Singh, please go ahead and add them to the chat. I know we have one comment from Gigi Macarivi, who said, wow, this is amazing and so informative. Thank you for all your hard work, Dr. Singh, and amazing collaboration and trust in our youth. So I would just echo that. I know it's great when we can get our younger folks involved and get them excited about science and get them involved in working on problems that are